citric acid cycle, a 4-carbon compound oxaloacetate, which is one of the key substrate of first reaction of the citric acid cycle, condenses with a 2-carbon compound formed from pyruvate dehydrogenase complex in order to convert into a 6-carbon citrate molecule. The citrate formed here will undergo isomerization and then oxidative decarboxylation. Isocitrate, by undergoing oxidative decarboxylation, it gets converted to 5-carbon compound alpha ketoglutarate. The 5-carbon compound alpha ketoglutarate again undergoes oxidative decarboxylation to yield a 4-carbon compound succinate. The succinate formed from alpha ketoglutarate undergoes a series of reactions in order to convert back to oxaloacetate, which is very important again to involve in the next round of TCA cycle further. The 2 carbon atoms entering the cycle as an acetyl unit are released or leaving the cycle in the form of 2 molecules of carbon dioxide liberated from oxidative decarboxylation of isocitrate and alpha ketoglutarate steps. As we know, in the molecules, energy yielding molecules, the energy is stored in their hydride ions. So, we extract the energy in the form of hydride ions from those molecules into the NAD plus and FAD. In the TCA cycle, at 3 places, at 3 steps of TCA cycle, there is formation of NADH, and at 1 step of TCA cycle, there is formation of FADH2. The hydride ions present in these compounds made or like participating in the TCA cycle, those will release the hydride ions into the nicotinamide adenine dinucleotide. The function of the citric acid cycle is the harvesting of high energy electrons from carbon fuels, as these electrons contain energy. In order to extract energy present in the hydride ions of these carbon fuels, we need acceptors such as NAD plus and FAD. NAD plus and FAD, they bind to these hydride ions and extract the energy from these carbon fuels, various carbon fuels taking part in cellular respiration and citric acid cycle. In the citric acid cycle, there is generation of 3 molecules of NADH and 1 molecule of FADH, which are sent to the electron transport chain. None of the chemical reactions of TCA cycle requires oxygen as a reactant. Neither we come across a step in the TCA cycle generating ATP directly, or large amounts of ATP are also not generated within the TCA cycle. Only 1 GTP molecule is synthesized. How is citric acid cycle helpful for cellular respiration then? The citric acid cycle instead removes electrons having the energy from these molecules, carbon molecules, into the NADH and FADH2 forms. Further, these 2 molecules, NADH and FADH2, are sent to the electron transport chain. When these NADH and FADH2 molecules reach out to electron transport chain, they give away these electrons to the electron chain complexes and runs the electron transfer across the electron chain complexes. Finally, these electrons are accepted by oxygen attached to the 4th complex of electron transport chain. So in order to continuously run this electron transfer within the electron transfer chain, the oxygen attached to the fourth complex plays an important role, as it is there to accept the electrons coming into the electron transport chain. Simultaneously, the electron transfer also establishes a proton gradient across the inner mitochondrial membrane. When the electrons are transferred between these electron chain complexes, the protons are pumped into the mitochondrial membrane space. These accumulated protons within the mitochondrial membrane space, they flow into the oxidative phosphorylation pump and helps in making ATP in the oxidative phosphorylation pump. That's how ATP is synthesized at the end of the cellular respiration. In any case, if there is oxygen deprivation in the cell and at the fourth complex of electron transport chain, it disturbs the electron flow. When the electron flow is disturbed between these electron transport chain complexes, it cannot establish proton gradient. No proton gradient, no flow of protons into the oxidative phosphorylation pump, results into no generation of ATP. So, the electron transport chain disturbance will not allow the acceptance of electrons from NADH and FADH2 further. So, the NADH and FADH2 accumulates in the mitochondrial matrix, indicating there is enough energy. As to run the TCA cycle, we require NAD and FAD molecules in order to extract the energy from the carbon compounds formed in the TCA cycle. As there is lack of NAD plus and FAD cofactors, the TCA cycle reactions cannot be taken place. It results into the decrease in the speed of TCA cycle henceforth.